All right. Good morning, friends. So good to have you back. I hope you had a wonderful holiday season and a happy new year.
Yes, good morning to everyone. Thank you. I'm actually really excited to get back onto the Field Trip Fridays horse.
Absolutely.
So shall we get started?
Yeah, let's do this thing.
All right. So today's field trip, we are going to the Giulio lab at the Nicholas School of the Environment for Duke University. And today we have with us Lindsay, who works in the Giulio lab. Hi, Lindsay. Thank you for joining us today.
Hi, everyone.
And each Friday, we have a DPS classroom host us. And maybe one day your school will host us too. But today from Moorhead Elementary, we have Miss Harden's Upper L class with us. So welcome, Moorhead Elementary. So glad you are here with us.
I'll share a
and then
picture
also
of
looks
Moorhead.
like Melissa from the Giulio lab has joined us too. Good morning, Melissa.
There's Moorhead. And it looks like Melissa's still getting her audio connected. Oh, there she is. Yeah.
Morning, Melissa. So to all of our friends in the chat, feel free to make any observations or ask any questions. Myself, Steve, Melissa, and Lindsay will be joining you in the chat to answer your questions. And let's see what this field trip has to do.
Yeah. Okay. I'm excited to show the video, so I'm gonna go ahead and share. All right. Looking good. All right.
I'm Jenna.
And I'm Steve.
Welcome to another Field Trip Friday. Where are we today, Steve?
We're at the Nicholas School of the Environment at Duke University, and we're going to be meeting Melissa Chernick and Lindsay Jaspers, who work in the Giulio lab, which studies fish.
Whoa.
Yeah, they study fish that have been adapted to particularly polluted river in Virginia called the Elizabeth River.
Yeah, I'm familiar.
You're from Virginia.
I am from Virginia.
We're going to learn a bunch of cool stuff about how they approach scientific problems.
I can't wait for that.
It's going to be super fun.
Let's go take a look.
Let's go.
My name is Lindsay Jaspers, and I am a postdoc in Rich Giulio's lab in the Nicholas School of the Environment at Duke and part of Duke Superfund Research Center.
I'm Melissa Chernick, and I'm a research technician in Richard Giulio's lab as well as David Hinton's lab, both of which are in the Superfund Center at Duke University.
Lindsay, Melissa, thank you so much for allowing us to spend some time in your lab today.
Sure, of course.
Can you tell us a little bit about what you all do here at the Nicholas School?
We study populations of fish. So we have one fish that we work with is the Atlantic killifish that lives all throughout the East Coast. But one of the places where we're primarily interested in studying these fish is the Elizabeth River, which is a very polluted place in Virginia. It has decades of pollution, and there's fish that have adapted to that pollution, and they've been able to withstand that and live there successfully for decades and decades. And we're really interested in studying how those fish have been able to survive there and adapt to that pollution. So we study different aspects of how they've adapted, what sort of costs there are to being adaptive. So are they more susceptible to other stressors because they've adapted to this pollution? And then we also study fish like zebrafish, which are just lab species where we can have different strains that allow us to test all different types of proteins and responses to chemicals, and we have a lot of methods that have been specialized for zebrafish that have enabled us to test a variety of different things with those fish. Maybe you can add some more to that.
Like Lindsay said, we study Atlantic killifish. We go up to the Elizabeth River in Virginia to collect the killifish every summer. We bring them back to our lab, and we have a nice tank setup, aquarium setup for them to live in, and we will collect their eggs, their offspring, in order to do some very controlled laboratory testing. These sites were designated as Superfund sites by the US EPA.
And so that's super fun.
Superfund with a D, yes.
Not
Fun
super
with a D.
fun.
They're not super. We have fun when we go to these sites, but I wouldn't classify them as super fun.
Right.
Yes, but super fun. And what this is it was a measure that was put in place decades ago where industries that cause a lot of pollution put money into this super fund in order to study effects of that pollution and clean up those messes. And so it's a pool of money that the EPA or other agencies can pull from in order to clean up these sites.
So yeah, what is some of the benchtop stuff that you find yourself doing? Like probably prepping like various solutions and things that you use for your
Right, so
computation.
we have these are sort of, we have a big cabinet of chemicals, but these are a lot of our commonly used chemicals, and we have pipettes and instruments that we need. And then we have pH meter, a microbalance, which is a very sensitive scale, and what's called a dissecting stereo microscope.
Nice.
So we can put whole fish under here for measurements. So for example, we have a pair of calipers here if we want to measure fish length or girth, then we can do that. But any sort of preparation is done in this area. So when we do embryo exposures and we want to check on them every day to see how they're doing, how they're developing, then we will put them under this stereo dissecting microscope here. And we have a camera on the microscope, and it's attached to this computer here where we can take pictures of those fish.
Cool.
Very
Cool.
Oh, it's so cool to see that
There we
blood
go.
flow.
Okay.
Yeah.
So the eyes, the head, so he's wrapped around the back of the yolk, and this is his tail coming up right here. And then this is the heart, so the atrium and the ventricle. These fish have 2 chambered hearts instead of 4 like we have. So down here the blood is coming through what are called the yolk veins, to collect in the sort of this confluence. It's called the sinus venosus. And then it goes into the atrium, the ventricle, back through the body, and it loops around again.
Cool.
And so this is the egg envelope, or the chorion. That's the protective layer of the fish. Down here is the yolk. These little bubbles here are lipid or fat droplets, which the embryo, especially right after it's hatched, when it becomes a larvae, will use as an energy source for those first few days before it can catch its own food. So it'll be on the yolk sac about 3 days. Depends on how active the individual is and how they are using up their energy. But you sort of have a head-on view. He's looking out at you in this picture. And so his mouth is right here. And once the yolk is gone, some of this stuff will sort of migrate back into the abdomen and then develop a little bit more. So the liver is there. It will develop more into a mature liver. Same thing with the heart. But for all intents and purposes, he has all of the organs, at least in very early developmental stages, that he'll have as an adult here.
So these fish are what we sort of sometimes call a stay-at-home fish. If you like that, especially in these times, we know what stay-at-home means. And they pretty much stay in the same area for their entire lives. So where they are hatched is where they live. They only move about 30 or 40 meters is their home range, which means if you have a high level of pollution at a very specific area, the fish are there. They're not going to move out to a cleaner area. It's just not in their life history or their evolution to do that. And so you either adapt or you don't survive. And so it's what we call a very high selection pressure on the population. And that has caused these fish to adapt to these very highly polluted environments. And so we go and we collect fish from these populations at these very specific sites from these different companies. We bring them back to the lab and we try to figure out how they do that.
So this is the sump tank, or the water reservoir, and it moves up through these pipes here to the 2 test tanks. And the fish are in these chambers within the test tanks. And the tanks have pumps, temperature sensors, and oxygen sensors in them. So the water is coming from this water reservoir. You can see it pouring in here. It's coming through here and then back down to the reservoir. So it's constantly cycling in here. So we have fresh water that's oxygenated going through to the tanks here. And then within the tube, at the beginning here, you can see sort of the gray honeycomb-like piece. And that is a fan that we can turn to start moving the water if those are the tests we want to do. And then all of the cables go back here to the computer, which is taking the readings from those sensors.
Cool.
Right now, resting metabolic rate or oxygen consumption is what's being measured. But if we were to turn on the fans, we could take a lot more measurements.
It's
so things
basically
like the
they're
velocity,
standing on a treadmill but not moving.
right? Sort of like the stamina, or how long they can keep swimming before they get tired. That sort of stuff are more active metabolics. So these fish were exposed to flame retardants when they were embryos, but then they were grown up in clean conditions. So these are the equivalent of about 65-year-old humans. So,
Cool.
So what we try to do, and this is where collaborations with multiple labs as well within our Superfund research program or center comes in, is in those lines of evidence we look at what we call different levels of biological organization. So the whole animal, to an organ, to its tissue, to a cell, to genes. And if you look at these different levels and you find the connections or links between them, a full story starts to emerge, and you start to get closer to solving that puzzle.
Yeah, really cool. So we got to see several of your really cool spaces and some of the amazing tools that you use to kind of probe these questions. And it seems like it's really important that this is like not only do these fish adapt, but so does your lab. It's kind of like evolved and grown over time. New postdocs come in, new graduate students come in, they have their own projects, they have their own funding, you know, all sorts of things kind of affect where the storyline goes.
One good science question spawns 10 more science questions.
Right.
And so we always have something to answer in the lab.
Yeah, once we answered one question, it just created like 10 more questions that we could answer. So once we figure out, oh, well, these fish have adapted to pollution, well, how have they done that? And then we can ask, okay, well, now we've remediated these sites, how do they respond to that? And do they go back? And what are the costs and the trade-offs of adaptation? So once we're answering one thing, it just builds upon more and more questions.
And in the laboratory, we design experiments where typically we change one thing. Everything is the same, we have different populations, but we change one thing. We add our chemical mixture. But the environment is a lot more complicated and complex. So studies like Lindsay's, that's looking at temperature, add in another thing. So you have all sorts of stressors in your environment, temperature, as she said before, low oxygen, and when we look at things in combination, the questions become that much more complex. And so those will spawn even more questions in the laboratory. So it certainly keeps us busy.
Yeah, absolutely. It seems like the journey is never over.
It's never, and we could, yeah, it's never over.
Lindsay, Melissa, thank you so much for letting us join you today at your lab.
No problem.
No problem.
Thanks for coming.
No problem, back at you.
Yeah, thank you so much. It was awesome to hang out with y'all. And for those of you watching, we're going to have a live Q&A with Melissa and Lindsay right after this video. So stay tuned for the live Q&A. We'll see you there.
See you there.
Bye, everyone. Alrighty. It was super fun. Thank you again so much for inviting us to your laboratory. It was an incredible visit, if I do say so myself.
Yes, absolutely. And our chat was active and quite curious today. So Ms. Harden, would you like to ask some of your students questions?
Sure. One of the questions students had was about the fish themselves. So they asked a lot about what the fish ate, if they were omnivores or carnivores or herbivores, and then as well as when you're taking care of them, how long it takes to feed them, how many tanks you have. Yeah.
Those are great questions. So the fish are omnivores. And I saw a question about how many tanks you have there. So you got to see a little bit of that in the video. For our killifish, we keep them in nice, really big tanks because they're pretty big fish and they need a lot of space to swim around. And we have about maybe 15 of those in the lab. And our zebrafish, which are much smaller, can fit into smaller tanks. And we have probably like 100 of those tanks. There's a lot of them. And in terms of feeding them, it's pretty quick. We feed them twice a day and it takes us maybe like 30 minutes to feed all of them just because there's so many individual tanks. But yeah, Melissa, if you have anything to add to that.
Yeah, you saw the brine shrimp in the video. So that's their live food. We also feed them a dry pellet food. And it's just like you would feed your fish at home. You just sprinkle it on the water and they come up and eat it. We do a lot of our fish care is very similar to what you might do if you have an aquarium at home. We just do it on a much, much larger scale.
Great. We also had a question about catching the fish, where they were coming from. Is there a time during the year that you catch them?
Yeah, sure. So we mentioned the Elizabeth River, and I saw a question in the chat also about maybe specific sites. So the sites that we go to are former wood treatment plants. There are 3 wood treatment plants that started operating in the 1920s, And so they were polluting the river for quite a long time with creosote, which is a chemical mixture. It's a wood preservative, and it gets down into the sediments, and parts of it can float in the water. And so what we do is we go to these former sites, either right after they were active, so before they were shut down and made a Superfund site, or a little bit later. So we have a few sites, 3 main sites that we go to on the river: Republic Creosoting, Atlantic Wood Industries Creosoting, and Hess and Eppinger Russell, which was also creosoting. And they are very close to each other, within about a mile, 1 to 2 miles of each other. So we can collect a good number of sites in a relatively short amount of time. So we always go in the summer. We usually like to start in about May, sometimes a little bit earlier. And what we do is we look at tide tables, because we like to go during high tides, which will be during the new or the full moon, when the tides are up about 3 feet. And so we time when we throw our traps. You saw us throwing some of our traps at the beginning of the video, to that high tide, and the traps will stay in the water for about an hour. And then we'll pull the traps out, and you saw us sorting some fish, and we put them in coolers so that they're nice and safe on the way home. And then they come into our system. And so we usually collect around anywhere from May to October, but it depends on how many fish we catch and how many trips we have planned during that time. But often we'll make anywhere at least 2 or 3 trips during the summer.
Cool.
Yeah, and if there's time, there was a question just to a little bit more about what the pollution particularly is from these plants, and does it affect other organisms?
Yeah, so the chemical that we're primarily focused on that comes from that creosote that Melissa was talking about is a class of compounds called polycyclic aromatic hydrocarbons, or called PAHs, and they're really toxic to all different species. In fish, we see reproductive effects and effects in their immune system and them being able to reproduce. But it also affects other species that live in the rivers as well. And they can affect humans as well. That's why these sites were so prioritized and being remediated, because it affects more than just the fish that live there. It was a danger to the humans that were in or around the river as well.
That was a great question. I think we've got time for a couple more. If there were, it was super active. Thanks for being so curious, friends.
Yeah.
It's super cool when y'all have so many awesome questions and observations.
And thank you also for talking about.
Yeah.
Sorry, go ahead, Melissa.
Sure. I was just scrolling through the chat a little bit here. Somebody asked if fish die during our experiments. We try to make sure that doesn't happen because what we like to do is we like to do things as in what we call environmentally relevant. We want to make sure that what we do in the experiment is what these fish would actually encounter in the environment. And remember, these fish are living in these environments. And so we've been working on these projects for many years, and we've figured out the concentrations of these chemicals that most closely mimic what they would see in the environment and don't kill the fish. So if we end up killing the fish, we don't really have an experiment. We don't we can't see what happens in their development. We can't grow them up to adults to see later life effects of early life exposure and things like that. So and there's always some natural mortality in a population, even a very, very healthy population. So I'm not saying fish never die, but we try to carefully control things so that we can see what's happening in the live animal.
Awesome. I think I saw one more question, and you might have already answered this, but what was the lifespan of the, let's talk about the Atlantic killifish. What's the typical lifespan of an Atlantic killifish?
A couple of years, maybe 2-3 years.
Cool.
Yeah.
All right, my friends, thank you so much. Lindsay and Melissa, thanks again for talking to Miss Hardin's upper elementary class. And Miss Hardin, thank you so much for bringing your upper L kids to learn about some cool fish research at Duke. Thanks again to thanks again to everyone at DPS, including Willow Alston-Soha and Dr. Linda Targaryen, our science specialist, and one more time, Moorhead Montessori Elementary. Thank you so much for hosting us. Steve, what are we doing next week?
Yeah, so next week, we're going to be taking a trip to the Eno River, and we're going to explore the Eno River in both space and time. So we're going to start in Hillsborough, where we'll learn a little bit about some of the history of the indigenous peoples that inhabited the land on the Eno and still do. Then we're going to travel downstream towards Durham as we explore some of the historical and current uses of the river, and we'll, along the way, develop an understanding of why this river and the resources that it provides are so important and so precious to our communities through history and to this day. I'm super excited about it. It was really fun. Jen and I had an amazingly beautiful day with the Eno River Association. So look forward to that. And let's see, with that, I just want to say one more time, Happy New Year. Thanks for joining us again. And thank you, everybody. Thank you, particularly Lindsay and Melissa. It was so fun visiting y'all. And, you know, I just want to say first this time that Jenna, you're a great host.
Thanks, Steve. I love hosting with you. You're a great host too.
Cool. Well, thank you, everybody. We'll see you next Friday. Take care, friends.
Bye, everyone.
Bye.